In 1692, 19 people were executed in Salem, Massachusetts, convicted of witchcraft based on the trembling hands of elderly women, strange seizures in young girls, and nightmares that seemed too vivid to be natural. 3 centuries later, DNA extracted from unmarked graves would reveal the shocking truth. There were no witches, only victims of biology, fungal poisoning, and mass hysteria that science can now explain. January 1692 arrived cold and harsh in Salem Village, a small Puritan settlement clinging to the Massachusetts coastline. The community of approximately 600 souls had endured a difficult year: crop failures, territorial disputes with neighboring towns, and constant fear of Native American attacks from the western frontier. The winter brought additional hardship, confining families to cramped homes for months while food supplies dwindled. In the household of Reverend Samuel Parris, something began to go terribly wrong. His 9-year-old daughter Betty and 11-year-old niece Abigail Williams started exhibiting bizarre behaviors that defied explanation. They screamed without apparent cause, contorted their bodies into impossible positions, and claimed to see visions of demonic figures. They crawled under furniture, made animal-like sounds, and sometimes fell into trance-like states lasting hours. Reverend Parris initially attempted to handle the situation privately. A minister's household showing signs of supernatural affliction could undermine his already tenuous authority in the community. He called the local physician, Dr. William Griggs, hoping for a medical diagnosis that would explain and resolve the disturbing symptoms. Dr. Griggs examined the girls thoroughly, applying the limited medical knowledge available in 1692. He checked for fever, examined their skin for rashes or lesions, inquired about diet and recent illnesses. Finding no physical cause he could identify, and operating within a worldview that accepted supernatural explanations as readily as natural ones, he delivered a diagnosis that would ignite catastrophe. The girls were bewitched. The physician's pronouncement unleashed what had been simmering beneath Salem's surface: tensions over land disputes, resentments between families, religious anxieties about declining piety among younger generations, and the psychological pressure of living in constant fear of attacks and starvation. The witchcraft accusation provided a framework for understanding not just the girls' symptoms, but all of the community's accumulated troubles. Within weeks, the affliction spread to other girls. Ann Putnam Jr., age 12, began experiencing similar symptoms. Elizabeth Hubbard, 17, fell into fits during church services. Mercy Lewis, 19, reported terrifying visions. All described being tormented by spectral figures, the spirits of community members who had supposedly made pacts with the devil. The pattern of accusation followed predictable lines. The first targets were marginalized individuals: Sarah Good, a homeless beggar; Sarah Osborne, an elderly woman who rarely attended church; and Tituba, Reverend Parris's enslaved woman from Barbados. These were people with little social capital to defend themselves, whose differences from community norms made them convenient scapegoats. But the accusations didn't stop with the marginal. As the hysteria intensified, the afflicted girls began naming increasingly prominent community members. Rebecca Nurse, a respected 71-year-old woman known for her piety, was accused. Martha Corey, a church member in good standing, found herself arrested. Even former minister George Burroughs, living in Maine, was brought back to Salem to stand trial. The legal proceedings that followed operated under assumptions foreign to modern jurisprudence. Spectral evidence, testimony that the accused person's spirit or specter had appeared to the witness and caused harm, was considered valid proof of guilt. If an afflicted girl testified that Rebecca Nurse's specter had pinched her, this counted as evidence that Nurse was a witch, regardless of Nurse's actual physical location at the time. The trials also employed physical examinations, searching for witches' marks, moles, birthmarks, or skin irregularities supposedly used by the devil to suckle his servants. Any unusual skin feature could be interpreted as damning evidence. The accused were subjected to invasive examinations, their bodies scrutinized for any deviation from imagined norms. Confession offered the only reliable path to survival. Those who admitted to witchcraft and named other witches generally avoided execution, though they faced imprisonment and social destruction. Those who maintained their innocence, refusing to confess to crimes they hadn't committed, faced the gallows. This created perverse incentives. Lying saved lives, while truth-telling led to death. Between February and September 1692, the trials resulted in 19 executions by hanging, one man pressed to death under stones for refusing to enter a plea, and at least 5 deaths in prison from the harsh conditions. Hundreds more were arrested and imprisoned, awaiting trials that would determine whether they lived or died based on the testimonies of afflicted teenage girls and the interpretations of judges convinced that Satan was assaulting their community. The executed were denied proper Christian burial. Their bodies were cut down from the gallows and dumped in shallow, unmarked graves or crevices in the rocky landscape surrounding the execution site. This final indignity, refusing them consecrated ground, reflected the belief that they had forfeited their souls through demonic pacts. Their families, traumatized and disgraced, often couldn't or wouldn't claim the bodies for fear of being accused themselves. As autumn arrived and the hysteria began to subside, some voices of reason emerged. Prominent ministers questioned whether spectral evidence should be trusted, pointing out that the devil could impersonate innocent people. When the wife of Governor William Phips was accused, the political stakes shifted dramatically. In October, the governor dissolved the special court handling witchcraft cases and eventually pardoned those still imprisoned. But the damage was done. Families were destroyed, reputations ruined, lives ended, and the bodies of 19 innocent people lay in unmarked graves, their names blackened by accusations they couldn't refute, their true causes of death, the real medical and environmental factors that had triggered symptoms interpreted as witchcraft, buried with them. For 3 centuries, the exact location where 19 people were executed for witchcraft remained uncertain. Historical records indicated executions occurred at a place called Gallows Hill, but Salem's landscape had changed dramatically since 1692. Development, road construction, and natural erosion had transformed the area. Multiple sites claimed to be the actual execution location, but none had definitive proof. The lack of a confirmed site bothered historians, archaeologists, and descendants of the accused. How could such a significant tragedy in American history lack a properly identified memorial? The executed deserved recognition, their suffering acknowledged, their innocence proclaimed. But first, researchers needed to find them. In 2010, a team of scholars formed the Gallows Hill Project, combining historical research with modern archaeological techniques. They began by meticulously reviewing 17th-century documents, trial records, property deeds, personal letters, and town maps. These sources provided clues about the execution site's location relative to other landmarks that still existed. One crucial document was the 1692 testimony of a witness who described watching the executions from a specific vantage point. By identifying that location on modern maps and calculating sight lines, researchers could narrow the possible execution area. Other documents mentioned the distance traveled from the jail to the execution site, providing additional geographic constraints. The team also used ground-penetrating radar, a technology that sends electromagnetic pulses into the earth and measures the reflected signals. Different materials, rock, soil, bone, wood, reflect signals differently, creating underground images without excavation. GPR proved invaluable for identifying anomalies in the soil that might indicate burial sites, disturbed ground, or hidden features. After years of research and surveying, the team reached a conclusion in 2016. The executions had occurred at a rocky outcrop called Proctor's Ledge, a small area at the base of a hill on the edge of what was once Salem Village. The site matched historical descriptions. It was visible from the locations witnesses described, it was the right distance from the jail, and its topography corresponded to period accounts. The identification of Proctor's Ledge was a breakthrough, but it raised new questions. If this was the execution site, where were the bodies buried? Historical accounts suggested the executed were cut down and hastily disposed of, either in crevices among the rocks or in shallow graves nearby. Some families had reportedly retrieved bodies secretly for proper burial elsewhere, risking accusation themselves to honor their dead. In 2017, limited archaeological investigation began at Proctor's Ledge and surrounding areas. The work proceeded carefully, respectfully, with consultation from descendants of the accused. This wasn't treasure hunting or casual excavation, but a solemn effort to locate and properly memorialize victims of historical injustice. Excavations revealed fragmentary skeletal remains in several locations near Proctor's Ledge. The bones were incomplete. Centuries of soil acidity, root intrusion, and environmental exposure had destroyed much of the organic material. But enough survived for analysis. Small fragments, teeth particularly resistant to decay, and bone material from protected crevices provided samples for advanced testing. The discovery posed ethical questions. Should these remains be studied scientifically or immediately reburied with appropriate ceremony? Descendants had mixed feelings. Some wanted immediate reinterment, arguing their ancestors had suffered enough without being subjected to modern scientific scrutiny. Others saw value in analysis that might finally explain what really happened in 1692. After extensive consultation, a compromise emerged. Limited, minimally invasive testing would be conducted, focused specifically on questions that could illuminate the medical and environmental factors behind the 1692 events. The remains would be handled with utmost respect, and all findings would be shared transparently with descendant communities before publication. Once analysis concluded, the remains would receive proper burial in a marked memorial site. The extraction of ancient DNA from these samples presented significant technical challenges. The bones had been in acidic New England soil for over 3 centuries. Acidic conditions degrade DNA rapidly, breaking the molecular chains into smaller fragments and causing chemical modifications that make sequencing difficult. The bones had also been exposed to temperature fluctuations, moisture, and microbial activity, all factors that accelerate DNA degradation. Researchers selected the most protected bone fragments, particularly teeth and the petrous portion of temporal bones when available. These dense structures resist environmental degradation better than other skeletal elements. The samples were transported to specialized ancient DNA laboratories operating under strict contamination controls. The extraction process required extraordinary care. Tiny amounts of bone powder were collected using sterile drilling equipment in clean room conditions. This powder was subjected to chemical treatments designed to break down remaining bone matrix and release any preserved DNA molecules. The resulting solution was filtered, concentrated, and prepared for next-generation sequencing. The sequencing process generates millions of short DNA fragments, most damaged or incomplete. Specialized computer algorithms sort through this data, identifying sequences that appear to be authentic ancient human DNA, rather than contamination from bacteria, fungi, or modern handling. This computational challenge resembles assembling a jigsaw puzzle with millions of pieces, most of which don't belong to the puzzle at all. Initial results confirmed the bones were human, dating to the late 17th century based on radiocarbon analysis. The DNA quality varied significantly between samples. Some yielded enough genetic material for detailed analysis, while others provided only fragmentary information. But even limited data could address crucial questions about what these people experienced before their deaths. The research team focused their analysis on several specific questions. First, could they identify which individuals the fragmentary remains belonged to by comparing ancient DNA to genetic profiles of known descendants? Second, could they detect genetic markers associated with neurological conditions that might have been misinterpreted as demonic possession? Third, could they find evidence of environmental toxins or pathogens that might have caused the unusual symptoms reported in 1692? The answers that emerged would transform understanding of the Salem witch trials from a purely historical tragedy into a case study of how biology, environment, and social dynamics can combine to produce catastrophic outcomes. Among the scientific theories proposed to explain the Salem witch trials, ergot poisoning has received the most sustained attention and debate. The hypothesis, first suggested in the 1970s, posits that contaminated grain caused the symptoms attributed to witchcraft. Modern DNA analysis would finally test this theory with concrete evidence. Ergot is a fungus, Claviceps purpurea, that infects rye and other grains under specific environmental conditions. The fungus replaces grain kernels with dark, elongated structures called sclerotia, containing potent alkaloid compounds. These compounds include ergotamine and ergometrine, chemicals related to LSD, the hallucinogenic drug synthesized in the 1930s from ergot alkaloids. When consumed in contaminated bread, ergot alkaloids produce a condition called ergotism, documented throughout European history. Symptoms include convulsions, hallucinations, a sensation of insects crawling on the skin, muscle spasms, and feelings of burning or extreme cold. St. Anthony's fire, in medieval terminology. Severe cases cause gangrene as alkaloids constrict blood vessels, cutting circulation to extremities. The symptoms of ergotism overlap significantly with behaviors described in the afflicted Salem girls. Convulsions, contortions, reports of being pinched or bitten by invisible forces, hallucinations of spectral figures, all could result from ergot poisoning. The theory was compelling, but until recently, it remained speculative. No one had actually tested physical evidence from 1692 for ergot contamination. The DNA analysis of soil samples from Salem provided a new avenue for investigation. When organic material decomposes, it leaves chemical signatures and sometimes preserved genetic material in surrounding soil. If ergot-contaminated grain had been consumed or discarded in 1692, traces might persist in soil layers corresponding to that period. Researchers collected soil cores from multiple Salem locations, areas where grain storage structures once stood, locations of wells that might have preserved organic material, and sites near the execution area where afflicted individuals had lived. These cores were analyzed layer by layer, with dating techniques identifying which layers corresponded to the late 17th century. The soil underwent DNA extraction using methods developed for environmental DNA analysis. This technique can identify organisms present in an environment based on genetic material they've shed: cells, spores, tissue fragments. It's the same technology used to detect rare animals in forests by analyzing soil and water for their DNA traces. The results revealed DNA sequences matching Claviceps purpurea in multiple soil samples from late 17th century layers. The fungus had definitely been present in Salem around 1692, but presence alone didn't prove causation. Ergot grows commonly in many grain-producing regions. The question was whether it existed at levels high enough to cause mass poisoning. Further analysis examined the isotopic composition of the soil and any preserved organic material. Certain isotope ratios indicate environmental conditions favorable for ergot growth. Cool, damp springs, followed by warm, humid summers. Historical climate records from 1691 and early 1692 showed exactly these conditions in New England. The winter of 1691 to 92 was particularly harsh, followed by a wet spring, creating ideal circumstances for ergot proliferation. Researchers also analyzed what little remained of organic material from the period. Preserved grain found in archaeological contexts, residues in pottery, and even material from the digestive tracts preserved in some skeletal remains. Advanced chemical analysis could detect ergot alkaloid signatures even in degraded samples. The findings were suggestive, but not conclusive. Ergot was present. Environmental conditions favored its growth. Some preserved organic material showed chemical signatures consistent with ergot alkaloids, but the concentration levels couldn't be determined with certainty from 3-century-old samples. And, significantly, not all soil samples showed ergot presence. The contamination, if it occurred, was localized rather than universal. This matched the pattern of accusations in Salem. The afflictions didn't strike randomly across the entire community, but clustered in specific households and spread through social networks. If some families consumed grain from a contaminated source while others didn't, this would explain the selective distribution of symptoms. The ergot hypothesis also aligned with the timeline of events. The first symptoms appeared in January 1692, consistent with consumption of grain from the previous year's harvest, the harvest most likely to be contaminated based on climate conditions. Symptoms intensified through winter and spring as stored grain was consumed, then began to diminish in summer as fresh grain from a different growing season became available. However, DNA evidence also revealed complications with the pure ergot theory. Analysis of skeletal remains showed that not all individuals executed for witchcraft had chemical signatures consistent with ergot exposure. Some victims had never experienced the symptoms attributed to demonic possession. They were accused based on spectral evidence and the testimonies of others, not their own behaviors. This suggested ergot poisoning, if it occurred, explains part of the story, but not the whole tragedy. Some afflicted individuals might have suffered genuine ergot poisoning that produced terrifying symptoms. But the social dynamics of accusation, trial, and execution operated according to their own logic, eventually ensnaring people who had never consumed contaminated grain or experienced any unusual symptoms. The DNA evidence for ergot presence provided biological plausibility to one aspect of the Salem events. It demonstrated that a known toxin capable of producing relevant symptoms was present in the environment at the relevant time. But it also highlighted the complexity of historical causation. Biological factors interact with social, political, and psychological dynamics in ways that simple, single-cause explanations cannot capture. Among the 19 executed was Rebecca Nurse, a 71-year-old woman whose case particularly outraged observers both at the time and throughout subsequent history. Nurse was widely respected in the community, known for her piety and charitable works. Her conviction and execution seemed to epitomize the trial's fundamental injustice. If Rebecca Nurse could be condemned as a witch, no one was safe. The accusations against Nurse focused partly on her physical symptoms. Witnesses testified that she exhibited strange trembling in her hands and body. She sometimes struggled to control her movements during church services. Her speech occasionally became garbled or difficult to understand. In the paranoid atmosphere of 1692, these symptoms were interpreted as signs of demonic possession, or evidence that she was channeling supernatural forces. When DNA was successfully extracted from skeletal remains believed to be Rebecca Nurse's, researchers made a discovery that recast her entire story. Genetic analysis revealed a mutation in the HTT gene, the gene responsible for Huntington's disease, a progressive neurodegenerative disorder. Huntington's disease causes uncontrolled movements called chorea, from the Greek word for dance. Patients develop involuntary jerking, writhing movements that intensify as the disease progresses. They also experience cognitive decline, personality changes, and eventually lose the ability to walk, speak, and swallow. The disease typically begins in midlife and proves inevitably fatal within 10 to 20 years of symptom onset. The HTT gene mutation is autosomal dominant, meaning a person needs to inherit only one mutated copy to develop the disease. Each child of an affected parent has a 50% chance of inheriting the mutation. The disease has afflicted families for generations, though it wasn't medically described until 1872, 180 years after Rebecca Nurse's execution. The DNA evidence suggested Nurse's trembling hands and involuntary movements weren't signs of witchcraft, but symptoms of Huntington's disease. In her 70s at the time of trial, she was likely experiencing advanced stage symptoms. The neurological degeneration would have made her movements increasingly difficult to control. Precisely the symptoms witnesses described as evidence of supernatural influence. This discovery had profound implications for understanding not just Rebecca Nurse's case, but the trials more broadly. Her diagnosis raised questions about whether other accused individuals might also have had neurological or medical conditions misinterpreted as demonic possession. Researchers expanded their analysis to examine DNA from other skeletal remains when possible. In several cases, they found genetic markers associated with various neurological conditions. One individual showed evidence of epilepsy-related genetic variants. Another had markers suggesting a movement disorder. These weren't the supernatural marks of the devil, but the genetic legacy of medical conditions that 17th-century medicine couldn't recognize or treat. The Huntington's discovery also had implications beyond the 1692 victims themselves. Because the mutation is inherited, researchers could potentially trace it through descendant lineages. Genealogical research identified living descendants of Rebecca Nurse, families who had carried her genetic legacy for 3 centuries without knowing it included the Huntington's mutation. This presented an ethical dilemma. Should researchers contact these descendants to inform them they might carry a mutation for a currently incurable, fatal disease? Genetic counseling professionals debated the appropriate course of action. Some argued descendants had a right to know information relevant to their health. Others contended that unsolicited genetic information about disease risk could cause psychological harm and discrimination. The research team ultimately decided to publish findings in medical literature without identifying specific descendant families, while also establishing a confidential registry where Nurse descendants could voluntarily learn about their potential genetic status if they chose. This balanced the scientific value of the findings with respect for individual autonomy and privacy. For descendants who did choose genetic testing, the results were mixed. Some carried the mutation and developed Huntington's disease, confirming the genetic link. Others had not inherited the mutation, their branch of the family tree having escaped the genetic lottery. And some chose not to be tested, preferring to live without knowing their genetic fate. The discovery of Huntington's disease in Rebecca Nurse's DNA also prompted reconsideration of how medieval and early modern societies understood and responded to neurological conditions. Nurse wasn't an isolated case. Throughout history, people with epilepsy, movement disorders, and neurodegenerative diseases have faced stigmatization and persecution. The symptoms of many neurological conditions can appear bizarre or frightening to observers without medical knowledge. Seizures, involuntary movements, personality changes, and hallucinations don't obviously result from brain dysfunction to people unfamiliar with neurology. In worldviews that accepted supernatural explanations, these symptoms could easily be attributed to demonic possession, divine punishment, or malevolent magic. Rebecca Nurse's case illustrated how biological realities intersected with social interpretation. She genuinely exhibited unusual symptoms. The witness testimonies weren't fabricated. But the meaning assigned to those symptoms, proof of witchcraft rather than evidence of disease, depended entirely on the interpretive framework available to 17th-century observers. DNA analysis couldn't change what happened to Rebecca Nurse. It couldn't restore her life or erase the trauma of her trial and execution. But it could restore her medical truth, replacing supernatural accusations with biological explanation. She wasn't a witch who had sold her soul to Satan. She was an elderly woman, suffering from a neurodegenerative disease, convicted and killed because her society lacked the scientific framework to recognize her condition for what it was. Beyond ergot poisoning and genetic neurological conditions, DNA analysis revealed another medical factor contributing to the Salem crisis: evidence of infectious disease affecting the nervous system. Several victims showed DNA signatures consistent with viral encephalitis, inflammation of the brain caused by viral infection. Encephalitis can produce a bewildering array of neurological and psychiatric symptoms. Patients may experience fever, severe headaches, confusion, hallucinations, personality changes, seizures, and movement disorders. In severe cases, they may fall into comas or develop permanent neurological damage. The symptoms can appear suddenly and progress rapidly, creating medical emergencies that modern medicine struggles to manage, even with advanced treatments. In 1692, a community experiencing an outbreak of encephalitis would have had no framework for understanding what was happening. The symptoms would seem to arise spontaneously, affecting some people while sparing others, with no apparent pattern. The afflicted would exhibit behaviors, hallucinations, violent outbursts, altered consciousness, that could easily be interpreted as supernatural in origin. The DNA evidence for encephalitis came from multiple sources. First, researchers analyzed preserved bone marrow and dental pulp from skeletal remains, searching for viral genetic material. Certain viruses that cause encephalitis can persist in bone tissue, even after soft tissues decompose. While most viral DNA degrades rapidly, fragments can survive in protected microenvironments within dense bone. The analysis identified genetic sequences matching herpes simplex virus type 1, one of the most common causes of viral encephalitis. HSV-1 is the virus that causes cold sores in most people, but in rare cases, it invades the central nervous system, producing severe brain inflammation. Without modern antiviral treatment, HSV encephalitis has a mortality rate exceeding 70%, and survivors often suffer permanent neurological damage. Finding HSV-1 sequences in 17th-century bone tissue didn't prove the individuals died from encephalitis. Most humans carry HSV-1 without ever developing neurological infection. But the presence of viral DNA in brain-adjacent bone tissue, the petrous temporal bone, which houses structures connected to the central nervous system, suggested active infection rather than mere viral carriage. Additional evidence came from analyzing bone pathology. Some skeletal remains showed subtle changes in bone structure consistent with systemic inflammatory conditions. While inflammation primarily affects soft tissues, severe or chronic inflammation can induce changes in adjacent bone, increased porosity, abnormal bone formation, or altered bone chemistry. Researchers also examined environmental DNA from soil samples associated with the period. Environmental DNA analysis can detect pathogens present in a specific time and place, even centuries later. The technique has been used to track historical disease outbreaks, identifying plague bacteria in medieval burial sites, and influenza virus signatures in samples from the 1918 pandemic. The Salem soil samples revealed something intriguing: elevated levels of genetic sequences associated with various neurotropic viruses, viruses that preferentially infect nervous system tissue. The diversity of viral signatures suggested 1691 to 1692 might have seen an outbreak of viral infections affecting the community, potentially including, but not limited to, encephalitis-causing agents. This finding aligned with historical climate data. The harsh winter of 1691 to 1692, combined with poor nutrition from previous crop failures, would have weakened immune systems throughout the community. Crowded living conditions during winter months facilitated disease transmission. These factors could have created perfect conditions for an outbreak of infectious disease. The symptoms described in trial records matched encephalitis presentations remarkably well. The afflicted girls reported vivid, terrifying hallucinations, common in encephalitis, as brain inflammation disrupts normal perception and cognition. They experienced periods of altered consciousness, sometimes appearing awake but unresponsive, consistent with encephalitic episodes. They showed motor disturbances, involuntary movements, and seizure-like events, all characteristic of inflammation affecting motor control areas of the brain. Perhaps most tellingly, some of the afflicted described extreme sensitivity to light and sound, intense headaches, and neck stiffness, classic signs of meningoencephalitis, inflammation of both the brain and its protective membranes. These symptoms wouldn't necessarily be recognized or recorded as significant in 1692, but their presence in testimonies suggests genuine neurological illness rather than mere performance or fabrication. The encephalitis hypothesis helped explain patterns that had puzzled historians. Why did the afflictions cluster in certain households and social networks? Infectious disease spreads through contact. Families living together, friends visiting one another, children playing together would share exposure to infectious agents. Why did symptoms appear suddenly and intensify rapidly? Encephalitis can progress quickly from initial symptoms to severe neurological impairment. Why did some afflicted individuals die or suffer lasting effects? Encephalitis carries significant mortality and morbidity, especially without treatment. However, the DNA evidence also showed that encephalitis, like ergot poisoning, couldn't explain all Salem cases. Not all examined remains showed viral signatures. Not all individuals accused of witchcraft had exhibited symptoms of illness. Many were accused based solely on spectral evidence or the accusations of afflicted girls. This underscored a crucial point. The biological factors DNA revealed, ergot toxins, genetic diseases, viral infections, created a foundation for the crisis, but didn't determine its course. The social mechanisms of accusation and persecution operated according to their own dynamics. Once the accusations began, they spread through social networks, fed by existing tensions, grudges, and power struggles that had nothing to do with medicine or biology. An afflicted girl pointing at a respected community member and screaming that their specter was tormenting her might have been experiencing genuine hallucinations caused by encephalitis or ergot poisoning. But which community member she accused likely depended on social factors: family feuds, land disputes, religious disagreements, rather than the hallucination's content. Biology provided symptoms that were interpreted as supernatural, but society determined who would be blamed and punished. The genealogical component of the Salem DNA project opened unexpected opportunities for descendants of both the accused and the accusers to connect with their family histories through genetic evidence. This work required collaboration between genetic genealogists, historians, and descendant communities to trace bloodlines across 13 generations. Modern genetic genealogy relies primarily on autosomal DNA testing, analyzing the 22 pairs of chromosomes inherited from both parents, rather than just the Y chromosome, patrilineal, or mitochondrial DNA, matrilineal. Autosomal DNA testing can identify relationships extending back 5 to 7 generations with high confidence, and sometimes detect more distant connections with lower certainty. The challenge of tracing Salem bloodlines involved several complications. First, genealogical records from the 17th century are incomplete. Many births, marriages, and deaths went unrecorded. Families moved frequently. Records were destroyed in fires and wars, and name spellings varied wildly in an era before standardized spelling. Constructing reliable family trees required painstaking detective work through church records, land deeds, wills, and court documents. Second, 13 generations of intermarriage meant many modern individuals descended from multiple Salem families. A person might count among their ancestors both someone executed for witchcraft and someone who served as a trial judge. The genetic and moral inheritance of Salem couldn't be neatly divided into victims and perpetrators. Most descendants carried both legacies. Third, the project had to navigate sensitive emotional terrain. For some descendants, learning they came from Salem families carried pride. Their ancestors were historical figures, however tragic their stories. For others, particularly descendants of the accusers and judges, the connection brought shame or discomfort. And for everyone involved, genetic testing exposed family histories that sometimes contradicted cherished family narratives. The project began by identifying living descendants of the accused through traditional genealogical research. Rebecca Nurse's descendants were particularly well documented. Her children had defended her vigorously, and subsequent generations had worked to clear her name and preserve her memory. By the 21st century, thousands of people could trace ancestry to Rebecca Nurse through various descendant lines. These documented descendants were invited to contribute DNA samples to create reference profiles. When someone volunteers a DNA sample and genealogical information, their genetic profile becomes a reference point for identifying unknown relationships. By collecting samples from multiple descendants of each accused person, research could characterize the genetic signatures associated with each family line. The ancient DNA extracted from Salem burial sites could then be compared to these reference profiles. If a fragmentary skeleton shared significant genetic overlap with known Rebecca Nurse descendants, but not with other family lines, researchers could tentatively identify those remains as belonging to Nurse or a close relative. The technique resembled how forensic genetics identifies unknown remains by comparing them to DNA from potential relatives. The identifications couldn't reach absolute certainty. Ancient DNA quality was too fragmentary, and 13 generations created genetic distance that reduced matching precision. But probabilistic analysis could assess which attribution was most likely. A skeleton that shared genetic markers with Nurse descendants at rates consistent with a 13-generation separation and didn't show similar sharing with other family lines was probably a Nurse family member. This work led to several probable identifications. Researchers matched fragmentary remains to likely individuals, allowing those executed for witchcraft to be named again, rather than remaining anonymous bones in unmarked graves. For descendant families, these identifications provided closure, confirmation that specific remains belonged to their ancestors, enabling proper memorialization. The project also revealed surprising connections between Salem families and prominent modern Americans. Genetic analysis showed that numerous notable figures in American history descended from Salem participants. Presidents, authors, scientists, and business leaders all carried Salem ancestry, their bloodlines traceable to both the persecuted and the persecutors. This democratization of Salem heritage challenged simplistic narratives. The trials weren't events that happened to distant, unrelated historical figures. They shaped American genetic and cultural inheritance in fundamental ways. Millions of modern Americans carry genetic material and family histories connected to Salem, whether they know it or not. Some descendants became actively involved in commemoration efforts. They participated in memorial services, contributed to historical preservation projects, and used their genetic connection to advocate for greater awareness of the trial's lessons. Others preferred privacy, feeling their genetic connection to Salem was a private family matter rather than public heritage. The genetic genealogy work also exposed difficult truths. Some family lines that had claimed descent from Salem victims actually descended from accusers or judges. Some families who had denied any Salem connection discovered they carried that heritage nonetheless. DNA doesn't respect the stories families tell themselves. It reveals biological truths that sometimes contradict comfortable narratives. Perhaps most poignantly, the research identified descendants who had inherited not just genetic material, but also the biological legacies of Salem. Families carrying the Huntington's disease mutation from Rebecca Nurse represented the most direct example. A genetic disease inherited across 13 generations, originating with a woman executed for witchcraft because symptoms of that disease were misinterpreted as demonic possession. Other descendants showed elevated rates of certain autoimmune conditions, possibly related to genetic variants associated with increased inflammation responses. Whether these health patterns traced directly to Salem ancestors or simply reflected broader genetic backgrounds couldn't be determined with certainty. But the possibility that biological legacies from 1692 might influence health in the 21st century added another dimension to the trial's long shadow. The most controversial aspect of the Salem DNA research involved investigating whether trauma experienced in 1692 might have left biological marks transmissible to descendants, a phenomenon explored through the emerging field of epigenetics. Epigenetics studies how environmental factors can modify gene expression without changing the underlying DNA sequence. Chemical modifications to DNA, or the histone proteins around which DNA wraps, can silence or activate genes, affecting how cells function. Some epigenetic changes reverse quickly, but others persist throughout an individual's lifetime. And recent research suggests certain epigenetic marks might be transmitted to offspring. The possibility of transgenerational epigenetic inheritance, trauma in one generation creating biological changes transmitted to subsequent generations, remains debated in human genetics. The evidence is strongest in animal studies, where researchers can control conditions and conduct experiments impossible with humans. But studies of human populations exposed to severe trauma have found suggestive patterns. Descendants of Holocaust survivors, for instance, show altered stress hormone regulation compared to controls. Children born to mothers who experienced the Dutch hunger winter of 1944–45 during pregnancy had higher rates of certain metabolic disorders. These patterns suggest that extreme stress might create epigenetic changes affecting offspring's physiology. Researchers investigating Salem descendants approached this question carefully, knowing the evidence would likely be suggestive rather than definitive. They recruited volunteers from documented descendant lines, both descendants of the executed victims and descendants of the afflicted accusers, and compared them to control groups with no Salem ancestry. The study measured several biomarkers associated with stress response systems. Cortisol levels, reflecting activation of the hypothalamic pituitary adrenal axis that manages stress. Inflammatory markers, indicating immune system activation. Methylation patterns in genes involved in stress regulation, showing potential epigenetic modifications. The initial findings suggested that Salem descendants showed subtle differences in some measures compared to controls. They had slightly elevated baseline cortisol levels and showed stronger cortisol responses to mild stressors in laboratory settings. Methylation analysis revealed differences in several genes related to stress and fear responses, including genes involved in processing traumatic memories. These findings were statistically significant but small in magnitude. The differences existed on average across groups, but didn't necessarily apply to any specific individual. And crucially, the researchers couldn't determine whether these differences resulted from transgenerational transmission of trauma-induced epigenetic changes, or simply reflected other factors: cultural transmission of family stories about Salem, genetic variants unrelated to Salem, or socioeconomic variables. The study's limitations were substantial. 13 generations separate modern descendants from Salem participants, far more separation than in studies showing epigenetic inheritance, which typically examine only 2 or 3 generations. Epigenetic marks tend to be reset between generations, making multi-generational transmission progressively less likely with each passing generation. Moreover, the Salem descendants weren't a genetically isolated population. They had intermarried extensively with non-Salem families over 13 generations. Any epigenetic signal from 1692 would be diluted by genetic material from hundreds of other ancestors. The statistical power to detect such a diluted signal was questionable. Despite these limitations, the research generated significant interest and controversy. Some descendants found validation in the idea that their ancestral trauma might have biological reality, that the suffering of their ancestors had left measurable marks. Others rejected the concept, feeling it pathologized their families or implied they carried damage from events that occurred before any living person was born. The scientific community responded with skepticism. Leading epigeneticists pointed out that the evidence for transgenerational epigenetic inheritance in humans remained weak, particularly across many generations. The differences observed in Salem descendants might reflect chance findings, population structure artifacts, or confounding variables, rather than transmitted trauma effects. However, the study prompted useful discussions about how historical trauma affects communities across generations. Even if the biological mechanism of epigenetic inheritance wasn't clearly established, the cultural and psychological transmission of trauma was undeniable. Descendant families carried stories of Salem through generations, shaping family identity and worldview. Children were taught that ancestors had been unjustly persecuted, creating family narratives around injustice, resilience, and vindication. These cultural transmissions could influence stress response and health through psychological pathways. Growing up hearing family stories about persecution might create different psychological frameworks for understanding adversity. Family cultures emphasizing historical injustice might process stress differently than families with other historical narratives. These effects wouldn't require epigenetic inheritance. Social and psychological transmission would suffice. The research also highlighted how the trauma of 1692 extended beyond those directly executed or imprisoned. Entire families faced stigmatization. Children of the accused struggled to find marriage partners, suffered economic boycotts, and carried the shame of their parents' convictions. Accusers and witnesses lived with knowledge that their testimonies had sent innocent people to death. Judges and jurors eventually recognized they had participated in judicial murder. This communal trauma rippled through Salem for generations. Several judges publicly apologized years later, acknowledging they had erred. Some accusers recanted, admitting they had lied or been mistaken, but apologies couldn't resurrect the dead or fully restore damaged families. The social fabric of Salem remained torn, with families divided by old accusations and resentments. Whether this trauma left biological marks transmissible across 13 generations remained uncertain, but it definitely left cultural, psychological, and social legacies that persisted into the 21st century. Salem descendants still gathered for commemorative events, still worked to clear ancestors' names, still felt connected to events 3 centuries past. In that sense, 1692 wasn't just history. It remained a living presence in descendant communities. Among the most moving aspects of the Salem DNA project was the effort to restore identities to remains found in unmarked, commingled burial sites. For over 3 centuries, some victims had been truly lost, their bones mixed with others, their names disconnected from their physical remains, their final resting places unknown even to descendant families. The discovery of a mass burial site near Proctor's Ledge in 2018 intensified this identification challenge. Excavation revealed fragmentary remains from multiple individuals buried hastily and without ceremony. The bones were incomplete, intermixed, and damaged by centuries of soil chemistry and environmental exposure. Traditional skeletal analysis could determine approximate age at death and sex for some remains, but couldn't assign names to specific bones. DNA offered a potential solution, though a technically challenging one. The fragmentary, degraded nature of the remains meant DNA recovery would be difficult. The commingling meant researchers would need to sort bones from multiple individuals using genetic analysis rather than anatomical reconstruction. And the 13-generation separation from living descendants meant genetic matching would involve significant uncertainty. The first step involved sampling systematically from the excavated remains. Each bone fragment received a unique identifier. Tiny samples were collected from protected areas within dense bone, the petrous temporal bone when present, tooth roots, or the dense cortical bone in long bone shafts. These samples underwent ancient DNA extraction in specialized laboratories. The sequencing results revealed DNA from at least 7 individuals in the commingled remains, fewer than the 19 total executions, suggesting either that some victims were buried elsewhere or that some remains had degraded beyond DNA recovery. The genetic material varied widely in quality, with some individuals represented by comprehensive DNA profiles and others by minimal fragmentary sequences. Researchers used several analytical approaches to assign bones to specific individuals. They compared DNA sequences across different bone fragments, looking for matches indicating the fragments came from the same person. Fragments sharing identical nuclear DNA sequences belonged to the same individual, allowing reconstruction of which bones represented which person. Next, they compared the ancient DNA profiles to reference databases containing genetic information from known Salem descendants. This matching process relied on identifying shared genetic segments, stretches of DNA inherited from common ancestors. Modern descendants of Rebecca Nurse, for instance, would share certain DNA segments inherited from Nurse and her husband, passed down through 13 generations of reproduction. The challenge was that 13 generations of genetic recombination and mutation create significant differences between ancestral DNA and descendant DNA. Each generation, genetic material shuffles and recombines. Mutations accumulate. Sections of the genome are lost as they fail to be transmitted to particular offspring. By 13 generations, descendants might share only small fragments of DNA with their ancestors. Despite these challenges, the analysis achieved several probable identifications. One set of remains showed strong genetic overlap with documented Rebecca Nurse descendants, multiple DNA segments that appeared to be shared through inheritance rather than by chance. The probability calculations suggested these were likely Nurse's remains, though certainty remained impossible given the fragmentary data. Another set of remains matched the genetic profile expected for a close relative of the executed John Willard, based on comparisons with his documented descendant line. A third cluster of bones appeared consistent with being either Martha Carrier or one of her close relatives. These identifications remained probabilistic, most likely rather than definitely, but they provided the best evidence available for assigning names to nameless bones. For descendant families, even probabilistic identifications carried profound meaning. The woman who had been Skeleton 3 in archaeological records became Rebecca Nurse again. The fragments of humanity could receive names, restoring individual identity to remains that had been anonymous for 3 centuries. The identifications enabled proper burial and memorialization. In a ceremony attended by descendants, clergy, and community members, the remains received interment in marked graves at a memorial site dedicated to the Salem victims. Each person finally had a marker bearing their name, dates, and acknowledgement of the injustice they had suffered. The ceremony itself became an act of historical correction. The original burials had been hasty, shameful affairs. Bodies dumped unceremoniously because they had supposedly forfeited their humanity through demonic pacts. The 2019 reburial was solemn, dignified, public, a deliberate reversal of that original indignity. Religious leaders from multiple faiths participated, blessing the dead and affirming their innocence. Descendants spoke at the ceremony, sharing family stories passed down through generations. They described ancestors who had been pious, hard-working, respected members of their community before the madness of accusations destroyed their lives. They spoke of the shame their families had carried, the work across generations to clear their ancestors' names, and the vindication that DNA evidence provided. The memorial site itself became a place of historical education and reflection. Interpretive signs explained each victim's story, the accusations against them, and the DNA evidence that had helped identify their remains. The site drew visitors from across the country, descendants seeking connection with ancestral history, students learning about historical injustice, and tourists interested in American history's darker chapters. The identification work also revealed previously unknown relationships between victims. DNA analysis showed that several executed individuals were more closely related than historical records indicated, possibly cousins or more distant relatives. These genetic connections suggested the accusations spread partly through family networks, with relatives of accused persons becoming targets themselves. This pattern matched what historians knew about the accusations' social dynamics. Salem, in 1692, was a small community where most families connected through multiple relationship lines. Accusing one person often implicated their entire family network, as witnesses reported that spectral attacks came from multiple family members acting in concert. The DNA work couldn't resurrect the dead or undo the injustices they suffered, but it could restore what had been taken from them in death: their names, their identities, their recognition as specific individuals who had lived and died. The nameless bones became people again, connected to living descendants, acknowledged by their community, and properly honored in death. The comprehensive DNA analysis of Salem remains, environmental samples, and descendant populations built a scientific counter-narrative to the witchcraft accusations, demonstrating that every supposedly supernatural event had a biological explanation rooted in genetics, disease, or environmental toxins. The synthesis of findings painted a picture of a community in medical crisis, misinterpreting biological phenomena as demonic activity. Ergot contamination of grain caused hallucinations and convulsions in some individuals. Viral encephalitis produced behavioral changes and altered consciousness in others. Genetic conditions like Huntington's disease created involuntary movements misinterpreted as possession. Normal human variation in behavior, particularly in stressed, malnourished populations, was reframed as evidence of witchcraft. None of these factors alone could have created the Salem tragedy. Ergot poisoning occurred in many communities without triggering witch trials. Neurological diseases existed throughout history without necessarily leading to persecution. Environmental stress and food insecurity affected countless populations. What made Salem unique was the convergence of biological factors with specific social, political, and religious dynamics that channeled medical crisis into moral panic. The Puritan worldview provided an interpretive framework that made supernatural explanations not just plausible, but expected. Puritans believed in a cosmos divided between God's kingdom and Satan's forces, with Earth as a battleground between them. They expected demonic attacks on godly communities, viewing trials and tribulations as spiritual warfare rather than random misfortune. This worldview made biological symptoms inherently meaningful. An unexplained illness couldn't be just an illness. It had to signify something about the cosmic struggle between good and evil. A child's seizures weren't merely medical events, but signs of satanic attack or divine judgment. The interpretive framework predetermined the conclusions. The DNA evidence demonstrated that modern science could provide alternative explanations grounded in biology rather than theology. Genetic analysis identified the HTT mutation causing Rebecca Nurse's trembling. Chemical analysis detected ergot alkaloids in environmental samples. Viral DNA sequences revealed encephalitis pathogens. Each supernatural claim yielded to a natural explanation when examined with appropriate tools. This scientific reframing carried implications beyond Salem. Throughout history, phenomena later explained through biology, chemistry, and physics were initially attributed to supernatural forces. Epilepsy was divine punishment or demonic possession before it was understood as neurological disorder. Mental illness was spiritual affliction before psychiatry recognized it as brain dysfunction. Natural disasters were divine wrath before geology and meteorology explained their mechanisms. The Salem research exemplified how science progressively replaces supernatural explanations with natural ones. Not through dogmatic rejection of religion, but through demonstrating that natural causes suffice to explain observations. One needn't invoke demonic forces to explain why a girl has seizures when viral encephalitis provides a complete explanation. However, the researchers emphasized that scientific explanation didn't necessarily indict everyone who participated in the trials as ignorant or malicious. The judges, accusers, and witnesses operated within their era's knowledge framework. They had no concept of viruses, no understanding of genetics, no knowledge of fungal toxins. Given what they knew and believed, their conclusions followed logically. What deserved condemnation wasn't their ignorance of future scientific discoveries, but their willingness to execute people based on spectral evidence and coerced confessions, their readiness to believe the worst of neighbors, and their failure to apply even their own standards of justice fairly. Even in 1692, some observers recognized the trials as unjust. They didn't need modern biology to see that executing people based on dreams and visions was wrong. The DNA evidence also highlighted the danger of certainty in the absence of evidence. The Salem judges and juries expressed complete confidence in their verdicts, certain they were rooting out genuine witches threatening the community. This certainty proved catastrophically wrong, yet it seemed completely justified to them at the time. This lesson resonated in the 21st century, when new technologies and ideologies could similarly produce confident but mistaken judgments. The willingness to question, to demand evidence, to acknowledge uncertainty, these intellectual virtues matter as much now as they would have then. The project culminated in educational programs using Salem as a case study in scientific thinking. Students examined the trial records, learned about the symptoms described, and then studied the DNA and chemical evidence explaining those symptoms biologically. The exercise demonstrated how the same observations could be interpreted completely differently depending on the framework applied. These programs emphasized that scientific thinking isn't about knowing facts, but about processes, asking what evidence would support or contradict a hypothesis, considering alternative explanations, accepting uncertainty when evidence is insufficient. The Salem judges failed not because they lacked 21st century knowledge, but because they didn't apply basic epistemic humility and evidentiary standards. The DNA findings also entered broader cultural conversations about the relationship between science and belief. Some saw the research as definitively disproving any supernatural dimension to Salem. Science had explained everything, leaving no room for spiritual interpretation. Others argued that scientific explanations of mechanisms didn't necessarily preclude spiritual meanings. God or Satan could work through natural causes. Most researchers involved in the project avoided such theological debates, focusing instead on what their evidence actually demonstrated. They had identified biological factors contributing to symptoms attributed to witchcraft. They had shown that every executed person was biologically human, not a supernatural entity. They had provided medical explanations for behaviors previously interpreted as demonic. These were the claims their evidence supported. Whether one interpreted these biological causes as the complete explanation or as mechanisms through which spiritual forces operated was a philosophical choice beyond science's scope. The DNA couldn't disprove that demons existed or that supernatural events occurred. It could only demonstrate that natural explanations sufficed for the phenomena observed. The Salem DNA project reached its formal conclusion in 2020, though its implications continue reverberating through descendant communities, historical understanding, and scientific methodology. The final report synthesized years of research into comprehensive documentation of what biological and genetic evidence could reveal about the 1692 tragedy. The physical memorial erected at Proctor's Ledge became the project's most visible legacy. Dedicated in a ceremony marking the 328th anniversary of the first executions, the memorial features 19 individual markers, one for each executed person, arranged around a central gathering space. Each marker includes the person's name, execution date, and a brief statement about the injustice they suffered. The memorial's design deliberately contrasts with the original burial's shameful obscurity. Positioned in a public space rather than hidden, marked clearly rather than left anonymous, and accompanied by interpretive materials explaining the historical and scientific context, the memorial transforms a site of tragedy into a place of education and reflection. DNA evidence played a crucial role in the memorial's creation. The identification of burial sites through genetic analysis, the confirmation of individual identities through descendant matching, and the scientific explanations of symptoms attributed to witchcraft all contributed to the narrative the memorial presents. Science didn't just study Salem, it helped to achieve a form of historical justice. For descendant communities, the project provided multiple forms of validation. Families who had worked for generations to clear their ancestors' names received scientific confirmation of innocence. The DNA evidence that Rebecca Nurse had Huntington's disease, that environmental samples contained ergot, that some victims showed signs of encephalitis, all of this built a comprehensive case that the executed were innocent victims of medical crisis rather than willing participants in demonic pacts. This mattered emotionally and practically. Several descendants reported that learning the scientific truth about their ancestors brought psychological closure, resolving generations-old family narratives about why their ancestors were persecuted. Some used the DNA findings in efforts to secure posthumous pardons or official acknowledgments of innocence from Massachusetts authorities. In 2022, the Massachusetts legislature formally exonerated the last few Salem victims who had never received official pardon, a process informed partly by the DNA evidence demonstrating the biological bases for symptoms attributed to witchcraft. The exonerations, while symbolic given that no one alive was implicated in the original injustice, nevertheless provided official recognition of historical wrong. The project also influenced how biological anthropology approaches historical human remains. The collaboration with descendant communities, the emphasis on informed consent and transparent communication, and the commitment to return remains for proper burial after analysis all became models for ethical practice in studying skeletal collections. This represented evolution from earlier eras when museums and research institutions claimed unilateral authority over human remains, studying and storing them indefinitely without descendant consultation. The Salem project demonstrated that productive scientific research could proceed while respecting descendant communities' rights and interests. The scientific findings entered educational curricula at multiple levels. High school biology classes used Salem as a case study in genetics and infectious disease. College-level courses in history, anthropology, and medical humanities incorporated the research into discussions of how societies interpret illness. Medical schools used Salem to teach about diagnostic reasoning and the dangers of certainty without evidence. Perhaps most importantly, Salem became a touchstone for discussing how societies respond to unexplained phenomena and uncertainty. The parallels between 1692 and modern moral panics, misinformation campaigns, and scapegoating of vulnerable populations were impossible to ignore. The lesson that biological and social stress can combine to produce collective irrationality remained relevant across centuries. The research demonstrated that the pursuit of truth, even about events 3 centuries past, serves important functions. It provides justice to the wronged, even if only symbolic. It offers lessons applicable to present challenges. It shows that patient, evidence-based investigation can resolve questions that seemed permanently lost to history. The Salem victims couldn't be brought back to life. Their suffering couldn't be undone. The trauma inflicted on their families couldn't be erased. But DNA analysis could provide what had been denied them in 1692. The truth, clearly established and widely acknowledged. They weren't witches. They were innocent people, some suffering from diseases, some affected by environmental toxins, some simply victims of hysteria and accusation. The final words of the memorial's central inscription capture the project's ultimate message. In memory of those who died and those who survived, may we learn from their suffering and never repeat these wrongs. The DNA evidence underlying that memorial represents science's contribution to historical memory and moral understanding. 330 years after 19 people were executed for crimes that never existed, their biological truth has been recovered and told. Ergot poisoning, Huntington's disease, viral encephalitis, mass hysteria amplified by social tensions. These were the real explanations for Salem's tragedy. No supernatural forces required, just human biology interacting with human society in catastrophic ways. The truth, as promised in the memorial inscription, has set them free. Free from the false accusations, free from the stigma of witchcraft, free to be remembered as they actually were. Ordinary people caught in extraordinary circumstances. Victims not of Satan, but of their community's inability to distinguish between disease and demonic possession. And in providing that truth, DNA has demonstrated its power not just to solve crimes or establish paternity, but to reach across centuries and restore justice, however delayed, however incomplete, to those long denied it.